Good day, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor, Metabolon, I'd like to welcome you to Metabolomic Approaches in NASH and NAFLD Research and Development. My name is Elizabeth Lamb, and I'm the host and moderator for today's event. Now I'd like to introduce our presenters for today. First is Dr. Carrie Wong, PhD, Senior Study Director for Metabolon. And our second presenter will be Dr. Jamie Bates, PhD, Research Scientist 2 for Gilead Sciences. So with that, I will pass the presenter ball over to you, Carrie. Welcome.
So today I'm going to talk to you a little bit about how metabolomics can be used in NASH and NAFLD research. Here's a quick agenda. We're going to talk about who Metabolon is, what metabolomics is, provide a brief NASH overview, and how metabolomics can be used for NASH research. And then I'm going to hand over the presentation to Dr. Jamie Bates at Gilead, where she's going to describe how she's used metabolomics in her own research. And then we're going to close up with how working with Metabolon and the different kind of approaches we can take. Metabolon was founded in about almost 20 years ago, and we're headquartered in Research Triangle Park, North Carolina, where we employ around 200 plus employees with expertise in anywhere from biochemistry, mass spectrometry, and software development. Our sole focus is in metabolomics, and this has resulted in a number of publications across a diverse set of areas in the life sciences. Metabolomics is really the basis of Metabolon's technology, and metabolomics is the study of the metabolites in the metabolome, which are a collection of the small molecules that are really circulating in every single living system. From our point of view, metabolomics allows us to directly assess the phenotype. So the metabolic signature is developed not just from internal cues such as DNA, RNA, and proteins, but also external factors, including diet, environment, and even the microbiome. And so since the metabolome is responsive to these internal and external cues, we find it's the ideal way to track and understand the connection of these influences to the phenotype. And in fact, metabolites have been used going way back in history, even to medieval times, where metabolites in urine provided a method to assess health. And then as we moved on and progressed into the 20th century, metabolites were used to understand how tissue function. And even in present day, single metabolite tests such as glucose are used in a clinical setting. And so these targeted approaches in traditional clinical assays such as glucose, cholesterol, and sterols can be very useful. However, with Metabolon's precision metabolomics technology, we can really open up the aperture and be able to screen for thousands of metabolites in one test. And this allows us to take a more holistic approach to studying the system or model of interest. And in this case, this system and disease model is NAFLD and NASH. And I don't have to tell this audience, but just to kind of reset, NAFLD and NASH is really the hepatic manifestation of the metabolic syndrome and results when there's an imbalance between energy expenditure and energy intake. And this energy surplus results in the deposition of fat in the liver. And in fact, it's estimated that approximately 60 to 80% of the obese population does indeed have fatty liver disease. And this disease is really a spectrum, a disease with a spectrum of severity, ranging from simple steatosis that we see in NAFLD, and then the onset and progression of fibrosis, cirrhosis, and potentially the development of cancer. At this moment in time, fibrosis does remain the primary clinical predictor of risk. And in fact, experts believe that in the coming 5 to 10 years, the leading cause of liver transplants will be this disease. And currently, liver transplantation is the only treatment. Studying NASH comes with its own set of obstacles, and these obstacles have been attempted to be met in a variety of different ways by different researchers. And one of the issues is that there are certain limitations in the tools that have been available in the past, and to a certain extent even today. And hopefully today I can convince you that metabolomics can be used to help really understand and study this disease. So I mentioned that there are numerous hurdles to studying NASH, and we'll get into the specifics of each one in the following slides, but the basic kind of obstacles are outlined here. So we have an unclear constellation of risk factors, and this is complicated by the fact that multiple molecular pathways are involved in the onset and progression of NASH. And then when we're trying to study all of these diverse inputs, we know that there is a lack of a robust preclinical model. So a lot of rodent models, one of the challenges is to understand if the rodent model does indeed replicate what we see in the human setting. And there's different approaches to developing clinical models. These include diet manipulation, genetic models, or a combination of the two. And later on in the talk, we'll discuss the different ways people have tried to identify appropriate preclinical models to study mechanism of disease. And then when we're also studying the disease in humans, right now the liver biopsy remains the gold standard for diagnosing and monitoring patients. And as you can imagine, this is not very practical. So a lack of an effective and practical diagnostic test or tool for monitoring patients with this disease is absent. The disease of NASH really envelops an unclear constellation of risk factors that are both diverse and occur over a long period of time, making the study even more difficult. So you have external inputs such as your diet, your lifestyle, the environment, your intrauterine environment, but you also have internal inputs that you don't necessarily have control of, and these include genetic factors, as well as family history, gender, and ethnicity. And then you also have the microbiome. And to compound the kind of multiple factors that can influence disease, you have the involvement of multiple molecular pathways that can be disrupted in NASH. And these include derangements in lipid metabolism, alterations in oxidative stress and excessive inflammatory responses, and microbiota metabolism. And as you see here in these figures, none of these pathways are mutually exclusive. And in fact, derangements, there's probably multiple parallel hits that occur in NASH, and that these multiple parallel hits create a vicious cycle of disease resulting in the progression. What we often see, and what a common theme of all of these different pathways and disease drivers, is the metabolism sits at the center of it and may serve as a functional integrator between these mechanisms. In addition to that, because of the uncertain and diverse disease drivers I just mentioned, this has resulted in a remarkable target diversity in which companies are currently trying to develop treatments for the disease. And most of these treatments involve metabolic processes in some ways or another. So by looking at and studying the metabolome, we can understand not just the primary causes and or consequence of these treatments and disease, but also secondary consequences. And that's really what's outlined here, is that the metabolism's role in NASH is very well accepted. And precision metabolomics allows us to interrogate a broad range of metabolites. So we know that energetics, lipid metabolism, cholesterol metabolism, inflammation, redox status, and mitochondrial function are all affected in this disease. And in one test, we are able to screen metabolites that are involved and play a role in each of these pathways. To that end, multiple groups are using metabolomics at various stages of the drug development process. Metabolomics has been used to identify targets and to verify target engagement at the discovery and candidate selection stage, and on through preclinical stages and clinical stages where metabolomics has been used to gauge response and identify unique signals such as baseline signatures of disease. So with that being said, I'm going to hand the presenter ball over to Dr. Jamie Bates from Gilead, where she will be able to describe how she's used metabolomics in her own research.
Great, thanks, Carrie. Appreciate the introduction. And as Carrie mentioned, here at Gilead, we're using metabolomics for a variety of experiments. I'm on the preclinical side, and I'm a biology lead for our acetyl-CoA carboxylase program. And so we generally in NASH employ two models to study disease progression, and this is because, as Carrie mentioned, no one model really represents the disease very well. So we like to use a high-fat, high-cholesterol mouse model for steatosis readouts, and then we use a choline-deficient, high-fat, high-cholesterol model for fibrosis readouts. And so our strategy on the preclinical side is to evaluate mechanism in these two models and also understand the MOA a little bit better with metabolomics and transcriptomics. Our colleagues on the clinical side are also using Metabolon platforms with two purposes. The first is to define targeted assays based on hypotheses that could be used for PD analysis or to measure the pharmacodynamics of your drug. And the second is to also do some exploratory work. In the clinic, of course, we're doing this with blood samples, and preclinically, we're doing this with liver samples. And the idea would be things that we identified as relevant to either disease or to treatment in the clinical samples, we can then validate in the blood in the animal samples. And so today I'm going to tell you about our experiments using acetyl-CoA carboxylase inhibitors in this high-fat mouse model. But we're also, as I mentioned, using the choline-deficient high-fat cholesterol model. And both of these are interesting because in the steatosis model, we see reductions in steatosis. But in the choline-deficient high-fat, high-cholesterol model, we don't see reductions in steatosis, but we do see reductions in fibrosis. So here, we're really interested in seeing what's happening to the lipidome in the presence of acetyl-CoA carboxylase inhibitor. And that'll come out in a later date. So today I'm going to focus on the steatosis model. Acetyl-CoA carboxylase is an essential enzyme in de novo lipogenesis. And what I'm showing here is that proof of concept that we dose a mouse with acetyl-CoA carboxylase inhibitor and we can repress de novo lipogenesis or the new synthesis of palmitate. So vehicle dose animals have 30% new palmitate synthesis over the time course. And then you can see that as you dose down, you start to see that the de novo lipogenesis comes back up to vehicle levels after 12 hours. And so this 1 mg per kg dose has partial target coverage over the 12 hours. So you'll see in the experiment that we include this dose to show partial coverage, and then we also include a higher dose that should be complete target coverage over the course of the experiment. So as I mentioned, the model is mice treated with what we call the fast food diet, and then treated from months 5 to 6 with either vehicle or acetyl-CoA carboxylase inhibitor. And I didn't mention before, but this acetyl-CoA carboxylase inhibitor is liver targeted. And all of the endpoints I'll be showing you have been measured in liver. We also include a control group that is age matched but fed a normal chow diet, and we're calling those the lean animals. And after a month of dosing, you can see that relative to the lean animals, the vehicle animals have significant steatosis, which is reduced with the partial target coverage and full target coverage of acetyl-CoA carboxylase inhibitor, and that's quantified on the left here as a % steatotic area. So we wanted to ask what was happening to lipidome in these animals. And Metabolon, we did the combination of the complex lipids and the HD4 platform, so the data will be intermingled here. So as I mentioned, acetyl-CoA carboxylase converts acetyl-CoA into malonyl-CoA, which feeds into the de novo lipogenesis pathway, and inhibition of it will block synthesis of palmitate, blocking DNL. And on the other side, malonyl-CoA can inhibit CBT1, and that CBT1 is responsible for free fatty acids being able to cross into the mitochondria and undergo beta oxidation. And so when you reduce malonyl-CoA, you relieve that inhibition and increase beta oxidation. And we've measured this with orthogonal methods. I showed you the de novo lipogenesis on the previous slide. So this data really just confirms that we're seeing a reduction in a marker of de novo lipogenesis. Palmitoleate is a little bit further downstream in the de novo lipogenesis pathway. And then 3-hydroxybutyrate is a product of beta oxidation. It's a ketone body, and you can see that as you repress acetyl-CoA carboxylase, those levels increase. So this was as expected. And we wanted to know what was happening to the individual lipid species. And here you can see that total triglycerides are up in the vehicle-treated animals relative to the lean control, and then are significantly down with treatment. And this is also true for the DAGs and the cholesterol esters and total cholesterol and total ceramides. Interestingly, while all of these lipid species are depleting, there are some that are increasing. And those include these membrane phospholipids, phosphatidylethanolamine and phosphatidylcholine. And this is of interest because it's been published that levels of these membrane phospholipids are reduced in the setting of NASH. And they are in our models as well. As you can see, the vehicle animals are reduced relative to the lean controls in gray. And then the levels of these lipids are increased, these phospholipids. And so then if you take a broader look of all the fatty acid species in all of the lipid classes. So here you can see each of the fatty acids in the columns and their enrichment in each of the lipid classes on the left here. And so what you notice right away is that the neutral lipids are predominantly increased on the diet. This top box is the vehicle over the lean. So this is telling you what's happening on the diet. And then below, you can see that with acetyl-CoA carboxylase inhibition, you have a repression of these. And for the most part, as you look across all of the species, those that are down with the diet seem to be increased with acetyl-CoA carboxylase inhibition, and vice versa. Except for in a few notable cases, there are some PUFA species that are down in the diet that are also still down, or down further in the ACC-treated animals. But that only exists really in the triglyceride species, which we thought was interesting. And I think what's interesting overall is just this seemingly reversal of everything that's happening, even in the context of still being on the diet, everything that's happening to the lipidome. Impressive to us. So we also wanted to see if there was any clue to what might be happening to mitochondrial function. There are definitely reports that mitochondrial function is impaired in the setting of NASH. And by NAD and ATP readouts, we see that to be the case too. And in our transcriptomic analysis, we had seen that treatment with ACC inhibitor increased expression of mitochondrial proteins representing all complexes. And so we were excited to see that signs of mitochondrial health improvement in the metabolomics as well. More interesting to us is that these changes were all occurring in what appears to be a state of less redox stress. So markers of oxidative stress include glutathione, which can be oxidized or reduced. And the reduced form is actually pretty difficult to measure accurately. And the oxidized form here, we were able to measure being reduced. And I can say that the reduced form, I guess I should say repressed. The reduced glutathione didn't change in this experiment, but the reduction in oxidized glutathione suggests improved redox status. Additionally, this is something that I learned from working with Carrie on the analysis. The increase in ophthalmate is actually an indicator that you have increased activity of this enzyme glutathione synthetase. So it's making ophthalmate, but it's also making glutathione. So that suggests that your capacity to make more glutathione, which improves redox status, is increased with acetyl-CoA carboxylase inhibition. So overall, it seems like when you're inhibiting acetyl-CoA carboxylase, you're able to completely revert the lipidome back to what it was before the diet, while improving mitochondrial function and reducing redox stress. Additionally, we saw some interesting effects on markers of bile acid synthesis. And so cholesterol itself is down with ACC treatment, and so are some intermediates of bile acid synthesis. And so we wanted to test if we had an effect on plasma bile acids. So in this synthetic pathway, the cholate is conjugated and released to the plasma as taurocholic acid. And if you look at measurements, this is actually done in a separate study of plasma taurocholic acid. We can see that ACC can significantly reduce these to a similar extent that an FXR agonist can. And so this is interesting to us. We would like to test further whether combinations of ACC and FXR might impact bile acid synthesis. So in conclusion, in this fast food diet model of NASH, acetyl-CoA carboxylase inhibition reduced steatosis and reduced markers of DNL and increased markers of beta oxidation. The liver lipid species that were increased on the fast food diet were largely reversed with ACC treatment, and likewise those that were decreased were increased for the most part, with a few notable exceptions, as I mentioned. And overall, in the context of changing all these lipid species, you're seeing improvement in oxidative stress status and mitochondrial function. And the suggestion that bile acid synthesis is affected by ACC is also something we'd like to follow up on. So oxidative stress is, of course, of great interest for NASH. So we're also following up by measuring markers of oxidative stress directly. And finally, as I mentioned, looking at combinations of FXR and ACC. So that's the end of my portion. Carrie, I'm going to pass the ball back to you.
Thanks, Jamie. That was an excellent example of how research in the preclinical setting has utilized and applied metabolomics in the study of NASH. I'm going to shift gears a little bit and kind of describe an example of how metabolomics has been applied in the clinical setting to understand NASH and how the potential of its utility in this space. So Metabolon has worked with multiple small cohorts, all really around 200, less than 200 patients. Then this has led to the potential identification of metabolic signatures associated with NASH. So in the example I'm going to provide to you today, this is a table of how the experiment or study was set up. NASH patients that were liver biopsy scored according to the NASH CRN criteria were recruited. There were about 50 patients with the fibrosis stage of mild, so that would be a stage of 0 or 1. There were 100 patients included in the intermediate or what can be considered an intermediate fibrosis stage of 2, and then 50 patients that had a fibrosis stage of 3 or 4. And what we did is that serum was collected and metabolomics was performed, and then we were able to take these metabolic profiles and correlate them to clinical outcomes. And in this example, it's fibrosis score, but in general practice, we can look at any clinical outcome or any endpoint of your choosing. So as you can see here is a list of, I've masked the specific metabolites, but revealed the superfamilies the metabolites belong to. And these are all the metabolites that we found that associated with fibrosis stage. And not surprisingly, if there's multiple pathways being affected by in this disease, you see multiple metabolites, a variety of metabolites identified. And these can be, obviously, not surprisingly, lipid and energy metabolism, but also amino acids and some cofactors and vitamins. Within this analysis, we found that metabolomics was able to outperform other minimally invasive methods of assessing liver fibrosis. So this is a ROC curve distinguishing fibrosis stage 0 through 2 patients versus fibrosis stage 3 through 4. And you can see the light blue line on the graph is depicting the metabolomics performance. Now this type of work really opens the doors in a couple of ways. It really kind of reveals the opportunity to incorporate metabolomics into diagnostic tools, something that's a minimally invasive diagnostic tool is really lacking in this area. And then in the short term, it also provides, gives us a tool to compare preclinical models to human models of NASH, something that's been mentioned multiple times throughout this webinar is the lack of correlation between preclinical rodent models of NASH and those in the human disease. So what we're able to do with our human cohort is to compare and contrast the metabolic signature from the human cohort to the preclinical models, and we can identify similarities. And this can be helpful in the identification of biomarkers or even mechanism and pathology of disease. And this takes us to working with the Metabolon and understanding that there's a continuum of capabilities. Ideally, we use our global metabolomics platform to really understand disease pathology, cast a wide net, and really use it as a hypothesis-generating tool in the assessment, in the identification of either target engagement or development of diagnostic tools. It's also a very convenient way to assess multiple pathways in one screen. Instead of having to do an experiment to study beta oxidation and then another one to amino acid oxidation and another one for redox status, this is all can be done in one fell swoop. And then we also have some more targeted assays, and these include the lipidomics assay, our complex lipid panel that Jamie referred to during her portion of the webinar, as well as a targeted assay team that can be helpful in developing clinical tools and refinement of those diagnostic tests. So to get a little bit more specific about how lipidomics can be used in tests and the advantages of our complex lipid platform is outlined here. So specificity is very important in being able to study lipids. Non-specific methods, for example, identifying phosphatidylcholine 36,2 provides you the number of carbons and saturation level of a lipid, but doesn't really allow you to map the species to the matrix outlined here. So the matrix outlined here identifies lipid classes across the top and the fatty acid species vertically. And by being able to visualize the complex lipidome in this way, we can easily identify patterns. So I mentioned specificity. You'll know that 16O The absolute value of 16.0 in the PC's lipid class. In addition, accurate quantitation utilizing a battery as standard allows us to sum not only rows but columns to, in fact, find total sums of lipid species. In addition, having complete coverage is important in really helping the interpretation. Now, at this point in time, a lot of the research is still catching up to the technology and understanding precisely what different species of fatty acids mean in the context of this disease. But I'm sure multiple researchers are investigating this, and this type of data is forthcoming. And then, as I mentioned, we have a targeted metabolomics team. And this team can be used in a variety of ways. So generally speaking, clients will utilize our global platform and identify 2 to 3 nodes of interest, of metabolic interest. And then these can be then narrowed down and isolated to focused assays developed to study these particular metabolites. And in fact, we do already have some off-the-shelf panels that can report on liver disease and could be liver function and can be useful in NASH. For example, we have a bile acid panel. We have, in addition to that, we have a panel that measures a bile acid precursor, 7-alpha-hydroxy-4-cholesten-3-one, and a cholesterol metabolism panel that could be of interest upstream of that. In addition to that, we have off-the-shelf panels that examine lipid metabolism, including a free fatty acid panel, a total fatty acid panel, as well as total triacylglycerides. And something that's, I think, of a lot of interest to multiple clients is our flexibility and our option to choose your own flavor. So if there's some metabolites that are of interest, you can speak to our targeted assay team, and they'll work with you to develop a validated assay to routinely measure a metabolic nodes of your interest. So with that, I will open up the floor to questions. If you don't already have a contact at Metabolon, feel free to call this number or email Carolyn Waite at the email listed here with any more specific questions regarding your research interests. Thank you.
Thank you very much, Carrie. We do have several questions that have come in. Our first question, how do you distinguish between metabolic perturbations that are causal in NASH or NAFLD versus those that occur as a result of the inflamed liver?
That's a tough question. That's a good question, and that's something that I think ongoing research is still trying to identify. One of the best ways to do it, and part of the complications of studying NASH, is that it's such a progressive and chronic disease that the ideal way to study that would be to sample either blood or liver at early or baseline, early, intermediate, and late stages of the disease, and then to follow the metabolic signature along with other endpoints and just to really understand which comes first in terms of the affected pathways.
Excellent. Our next question, what evidence do you have that demonstrates that murine models of NASH translate clinically?
Well, I think, so Jamie kind of touched on that, and they're using a combination of murine models in their studies, and that's something that that's why we offer the comparison between the human and the clinical metabolic human and mouse metabolic signatures, is to identify those models that could have the most utility in a clinical setting.
I can speak to that a little bit too. I think it's a great question and one of ongoing research. And from the acetyl-CoA carboxylase side, I can say that our model of steatosis was very representative. We dosed at a level that gave partial inhibition of acetyl-CoA carboxylase in the mouse, and we know that in the clinic, the dose we're using gives partial inhibition too from de novo lipogenesis labeling studies. And in both settings, we see an average reduction in steatosis of about 30% in the clinic by MRI-PDFF and in the mouse by both morphometric analysis, but also just by triglyceride quantification. So that gave us a surprisingly good translatability. The fibrosis side, I think, still remains to be seen. We have a lot of agents that you can see reduce fibrosis in rodent models, and we have yet to see something that is as potently effective on reducing fibrosis in the clinic, I guess you might say. So I think fibrosis models are where we need some more work, but the steatosis part, I think we have good models.
All right. A series of 2 questions. How is ACCI different from FASTNY? And then the follow-up question, how is ACCI targeted specifically to the liver?
Oh, sure. Yeah, I did answer those questions because those were sent to me, I thought, privately. But yeah, those are great questions. So the ACC inhibitor is the first enzyme in de novo lipogenesis, and then FASN, fatty acid synthase, is just the next enzyme downstream. So you would imagine that they both would do the same thing of inhibiting de novo lipogenesis, and they do. The difference is that the product of acetyl-CoA carboxylase is malonyl-CoA, and so when you inhibit ACC, that goes down. But when you inhibit the next enzyme downstream, FASN, malonyl-CoA goes up. And malonyl-CoA, if you remember, I went over it kind of quickly, is that inhibitor of beta oxidation. And so only the ACC inhibitor will reduce malonyl-CoA, relieving inhibition of beta oxidation, and thereby you get increased beta oxidation, which is a good thing because that's the burning of fat. And the second question about the liver targeting, is that right?
That's correct.
Yeah, the liver targeting is by making the compound a substrate of OATP transporters, which are hepatocyte-specific transporters. So that's how you make a compound liver targeted.
Okay, perfect. Our next question, what is the relationship between metabolomics and genetic-based risk factors for NASH?
So there's been some studies that have kind of examined that topic. I think Arun Sanyal had a paper a couple years ago where they looked at PNPLA3, which is a genetic variant that is implicated in the risk of developing NASH. And they found in that paper that in addition to its potential triglyceride hydrolysis work, you also saw evidence of kind of a catabolic response. So amino acid catabolism went up. And so that's just one example of how metabolomics, how a genetic marker was identified and metabolomics was used to really understand the global metabolic implications of that particular genetic variant in NASH.
Our next question, NASH in vitro models, are they available and translational to in vivo studies?
So right now, Jamie, feel free to add on. Go ahead. Feel free to add on, but generally speaking, each client that we've worked with kind of picks their own model, and so they'll choose a specific diet or genetic model based on the literature and their own preferences and goals, and that's how they will determine which model they're using. Jamie, do you have something else to add to that?
No, I think it's a great question. There are different companies that can make in vitro co-culture systems or organoid systems like Organovo or Infero. But we're still working on finding the ideal in vitro model. Of course, you can culture hepatic stellate cells to test antifibrotic activity. But right now, we still, mouse models are the golden rule. So still working on that.
All right. Our next question, what's the ideal study design look like to get the most robust insight?
That's also a good question. Well, one of the important things to do is to understand that you need to have a strong control group. So that's something that Jamie's study did quite well, in that we have not only a vehicle group that we can compare back to get our fold changes in metabolites, but we also have a lean group. So step one would be to identify a good control cohort. And then step two would be make sure that it's a properly powered study. So Metabolon has performed power calculations and identified the proper number of N depending upon the system being studied. So for humans, we recommend at least over 30 subjects per group, and that accounts for all the helps to account for the individual variability that you observe in humans. When we look at rodents, again, Jamie's was very well powered. She had 10, which is exactly what we often recommend to our clients. And then when we get down to cells, biological replicates of 4 or 5 can be used to identify metabolic signatures.
We've had a couple questions come in regarding the microbiome. What specific role do you see the microbiome playing? And does Metabolon's platform detect any specific biomarkers generated by the microbiome?
Yeah, so the microbiome is always a hot topic, especially in NASH and in many other diseases. Our platform detects many metabolites that can be linked back to microbial metabolism. Now, the research hasn't quite gotten to the point where I can say metabolite X corresponds with a shift from Bacteroides or firmicute populations, but we have detected changes and shifts in metabolites that do reflect shifts in the microbiome. So if you can combine 16S sequencing with metabolomics, you could potentially not just understand changes in the microbiome in NASH, but also begin to understand the relationship and interplay between the microbiome and the host metabolism. And so we do have a number of metabolites that are the result of tryptophan or aromatic amino acid metabolism. Secondary bile acids reflect a microbial metabolism, and we detect quite a few of those as well.
All right. Our next question also about the metabolome. Does the metabolome profiling of mice provide insights into what happens to the acetyl-CoA that doesn't go through DNL when ACC is blocked?
Yeah, I think that's a great question, and one that I unfortunately don't have a good answer to right now, but it's definitely something we're curious about. You might expect that increased acetyl-CoA could be a substrate for making cholesterol, but I showed decreases in liver cholesterol, so that doesn't appear to be happening. Other things that could happen that we haven't measured is acetylation of proteins or acetylation of DNA, but others have reported changes in those parameters. So that's something that we're still researching more. Carrie, I don't know if you wanted to take it all from your analysis.
Yeah, I think those are very good points. And also, you saw increases in the beta oxidation and increases in ketone body formation. So we measured one ketone body, BHBA, that was found to be elevated upon inhibition of acetyl-CoA carboxylase. And that's actually formed when acetyl-CoA levels are in excess. So a potential path forward could be to take a closer look at ketone bodies in the liver as well as in the in the circulation to understand as a potential pathway of acetyl-CoA catabolism or metabolism.
Brent says data presented at the liver meeting indicated that ACC inhibition led to hypertriglyceridemia in humans. Do the mouse experiments provide insights as to why this might happen?
Yeah, that's a great question. So in this mouse model, we did see increases in plasma triglycerides with the higher dose, which is the complete target coverage. But if you dose lower, we could avoid those increases in plasma triglycerides. However, in the clinic, it's not clear whether that's dose responsive just yet. But looking at transcriptomics, we can see a reduction in PPAR alpha signatures and an increase in SHRB-P1C signatures and LXR alpha signatures. So we think that you could be tipping the balance a little bit in that direction. And so one way to ameliorate that would be to treat with a PPAR alpha agonist. And this was also published in a recent Cell Metabolism paper coming out of Jay Horton's lab. So our mouse data agrees with that as far as the transcriptomics go. And I showed you in the metabolomics that we saw reductions with ACC in some of the PUFA species, specifically in the tags. But interestingly, from the HD4 platform, where you can actually distinguish the N3 from the N6 species, we did not see changes in those metabolites that are thought to be the best PPAR alpha agonists. So the hypothesis was that the PUFAs responsible for agonizing PPAR alpha are reduced. And we did see global reduction in PUFAs, but the specific ones that were PPAR alpha agonists, we didn't see reductions. But then if you go back to the data where I showed specifically in the triglycerides, some of the PUFAs were down, suggests that maybe those specific fatty acids, maybe it depends which lipid class they're in for how they can signal. But as far as the total PUFAs that should agonize PPAR alpha, we didn't see that those were reduced. So actually our metabolomics put a little bit of a wrinkle in that hypothesis. So then we don't know what could be tipping that balance. And unfortunately, we don't get a good read on levels of oxysterols because those could be agonizing LXR alpha, but we don't know yet if those levels are changing in the model. But we were able to validate what they showed in the Horton paper, that a PPAR alpha agonist can overcome the increase in plasma triglycerides induced by acetyl-CoA carboxylase inhibition.
All right, perfect. A question for both of you. Does Metabolon or Gilead have experience running plasma samples along with liver samples? And does Metabolon's platform work on cell-based experiments?
Yeah, yes. Metabolon, we can pretty much run any matrix that you have in your body or around your body. So that's any tissue, any biofluid, urine, blood, and cells as well, both supernatant and the media, as well as the cell extracts themselves.
Oh, I'm sorry, you asked specifically about Gilead. Yes, we've run both, and I think it's of use. Lately, I think the approach is more to use plasma for the clinical samples because that's what's available, and then we'll go back and validate that in our preclinical models, so as not to chase something that's not relevant in the plasma of the rodents.
Okay. And our final question, this would be for Carrie. When working with Metabolon, what's the process?
When you initially come and start to work with us, we'll sit down and we'll discuss your project goals and try to design a study that gives you the best opportunity to receive actionable data. Following that, we will receive your samples and we'll run them on our platforms to identify the metabolites. And so we use a library-based approach to identify these metabolites, and that allows us to generate a list of detected metabolites as well as their levels, and then we then use that list to statistically compare each study group to the control. And the kind of the deliverables that you'll receive will be not only that statistical readout as well as a list of the metabolites, but also a report and presentation where we will interpret your data and highlight the metabolic changes and their meaning to your system.
All right, perfect. Well, we're coming up onto the hour, so I'd like to very much thank our presenters for today, Dr. Jamie Bates and Dr. Carrie Wong. I'd like to thank Metabolon for sponsoring today. And most of all, I'd like to thank those of you who came and spent this time with us. We hope you gained some information that will help make your job a bit easier going forward. So with that, I'll wish you all a great day. Thank you. Bye-bye.